Good morning, good afternoon, good evening. Wherever you are or what time it is, welcome to UC Grad Slam. I'm Pamela Brown, Vice President of Institutional Research and Academic Planning, and really thrilled to be here today. 180 seconds, that's it. That's what these students here have got to be able to convey in a complex and cutting-edge research that they're working on to all of us so I can understand it. That's the fun and the challenge of UC Grad Slam. Now, you should know that every one of these students is already a winner. Each of them has competed on their campus, and they've been selected to come and represent their own alma mater today here. And we are really, really excited to get this showcase of UC innovation and graduate education and be able to see this now. I want to take this moment to recognize that academic graduate education is the foundation of the University of California's status as a world-class research university. UC is the nation's leading public academic research system and is the state's research arm for education in California. UC's role in training the next generation of researchers is the centerpiece of our mission. And our graduates are highly skilled and analytic professionals who drive the modern economy and help shape the world in countless ways. So I'm super excited that we get to showcase just a bit of what our students are doing and how their work impacts our society. Being the head of institutional research and academic planning, stats and facts, those are what I do, but I know in my heart, it's much more powerful to share that when you tell stories that matter, and that's what we're going to be hearing today. Whether you're watching online or you're here in the room at LinkedIn, we're super glad that you decided to slam with us. You're going to get to see the world of UC through the lens of a graduate research education. So buckle up. We're about to go on a journey and see firsthand an impressive array of impactful research taking place across the University of California. So I've got the pleasure of bringing on the person who's really going to get this presentation party started. Nathan Brostrom is a friend, and he's a wickedly smart advocate and leader for the University of California. Nathan serves as the Executive Vice President and Chief Financial Officer for the University of California, and he does all things financial. Whether it ends up being asset management, debt management, system-wide operating budget, capital finance and planning, risk services, procurement, financial accounting, this is basically Monday. In 2019-20, Nathan stepped up to serve as interim chancellor at UC Merced.
Go Bobcats.
And experience. That's really good. My goodness. Yeah. An experience that I know both he and the campus really loved. But Merced isn't Nathan's only campus experience. Before coming to the Office of the President, Nathan served as Executive Vice President for Business Operations and the Vice Chancellor for Administration on the UC Berkeley campus. That was really, I mean. Go Bears! All right. So he's worked at the youngest campus with a lot of energy and the oldest campus that we're going to wake up today. And he cares for every other UC campus in between. Now, while Nathan is a graduate from Stanford, I know. Insert pause.
Pause.
And he also got his master's at Princeton. He is one of the greatest advocates for public education and especially the University of California. And I know he's thrilled to be here today. It is my honor. And I ask you to join me in welcoming Executive Vice President Nathan Brostrom.
Pamela, and it is so great to be here. This is one of my favorite events of the year, so welcome all of you to Grad Slam 2024. Pamela did not mention that one of our frequent interactions is we're both hardcore Warriors fans. And often watching a Warriors game, I will text her a question, And by the fourth quarter, she has it back. She's got all the facts and statistics on her in her mind. I'm so pleased to be here today, and we are going to hear from some of the brightest minds in our overall university. And we can't wait to hear about your research. I also want to thank our gracious host, LinkedIn, for once again providing space and support for Grad Slam. We're grateful for your partnership. President Drake really wanted to be here today, but we've got quite a few things going on, and so he asked if I would come and pinch hit, which was much easier than being on a special regents meeting right now. Especially because this is a really joyful event. It really is a celebration of our amazing graduate students and one of the most important missions of the university, the pursuit of new knowledge and the sharing of discoveries with the world. One thing that Pamela did not say is that our overall sponsored research is about $7 billion. We get $5 billion from the state of California, which we're very grateful. We get more money from the federal government through NIH and NSF and all of the research arms. So the 10 graduate students we are about to meet are already champions. They've won their campus competitions at explaining their research in ways that both engage us and make it understandable. And as winners of their campus Grad Slam contest, they've already proven their skills as both scholars and communicators. So we're all in for a great treat today. I now want us to meet our judges. This distinguished panel has the important job of assessing how well each participant engages the audience and communicates their research. I'm going to ask each of the judges to briefly introduce themselves, both to our in-person and virtual audience. So let's begin with Professor Chu.
Hello, I'm so excited to be here. My name is Diana Chu, and I am a product of the UC system. Go Bears, go Bruins. I promise that won't bias me. I am currently a professor at San Francisco State University in biology, where I study how sperm is compacted in sperm. That's important for fertility. And I also teach a scientific communication class at San Francisco State, and that you can see the 3-minute thesis talks for SFSU students at SFSU SciComm. Thank you.
Good morning, everyone. My name is Nancy Lee, and I have the honor and privilege of serving as a member of your UC Board of Regents. Thank you so much for having me this morning. I'm really excited to be here. I'm very much looking forward to this event. In particular, I'm really looking forward to learning about how each campus representative is thinking through very challenging and complex issues, but in innovative ways, and that will have real world impact. So, these types of events are such great reminders as to why our UC system is so world class in a truly authentic sense. So thanks for having me, and I look forward to our discussion.
Good morning, all. My name is Daphne Jomo. I'm a published author. I'm a student of San Francisco State, majoring in computer engineering. I'm also a Cowbridge scholar, which is why I'm here today. A Cowbridge bridges a gap between the CSUs and the CUs. They create opportunities for underrepresented minorities and communities to take part in PhD programs in physics and astronomy, maths, computer science, and computer engineering. It's a great program, and over the course of the academic year, we attend workshops on different UC campuses. We attend conferences to prepare students ahead of their PhD goals. It's a very great opportunity, and I'm glad to be here today. Looking forward to all the research presentations, and I thank you all for having me.
Morning, everyone. Jim Steintrager. Like Nathan, I am not a UC product. I went to Columbia for my PhD, so it's been really interesting watching the news for the past week or so. I am a professor of English and comparative literature at UC Irvine, where I've been for zot. Make sure you get the finger back, or else it's a satanic goat. I've been there for 25, 26 years now, so some UC experience at this point, and I am currently the chair of the UC System-Wide Academic Senate, so faculty representative for all 10 of the campuses. Thank you.
Hi, I'm Stephanie Wiseman. I'm the artistic director and founder of The Marsh, a breeding ground for new performance, which is a theater, not a swamp. Somebody shake their hand. There's swampy aspects and there's all kinds of aspects. I'm so glad to be here today. I'm from State University of New York at Buffalo.
Mm.
All right, SUNY Buffalo. Undergraduate, graduate, and I was thinking about my science, like why am I here? But thinking about my science, I guess it's the communication, and I would like you to know that my circulatory system that was made out of clay in 4th grade won. However, the DNA plaster Paris thingies I made in 5th grade lost. I'm really glad to be here and can't wait to see it all. Thank you so much.
Thanks to all our judges. We really appreciate you taking the time here. I do know I traveled with Jim last week. And he told me he's writing a book on the Marquis de Sade. So we may ask him to do a 3 minutes on Marquis de Sade over lunch. Yeah. But in addition to this group, there is one more very special judge participating in Grad Slam today. That's all of you. Once again, this year, we will give out a People's Choice Award that will be determined by your votes. So after today's presentations end, we will provide instructions on how you can text to vote for your favorite presenter. So please make sure to keep track of the talks and vote after they're all done. So now, without further ado, let's get on to Grad Slam. The research these graduate students will talk about today represents years and years of hard work. From wherever you are, please give each of our contestants a warm welcome as they come up on stage. So now I'd like to welcome our first contestant to the stage, Melis Chakar from UCLA. Welcome.
Have you ever thought about fashion in science? No, I'm not talking about designer lab coats or luxury brand beakers, but of course there are trends of research within science, and some topics gain popularity while others fall out of fashion. I'm a neuroscientist, and today I will tell you about a brain region that fell out of fashion for the wrong reasons and is now making a comeback. This brain region is called the cerebellum. The cerebellum helps you perfect your fine motor skills. So let's say you want to play a note on the piano. You place your hands on the keyboard, and at this point, your cerebellum has a sensory prediction, or a prediction of what that note will sound like. But something goes wrong, and your hand is not in the right place, and when you play it, the note doesn't sound right. You know that you made an error, and your cerebellum knows that you made an error. Your cerebellum then communicates this error to the rest of your brain, so that next time you want to play this note, you put your hand in the right place. So it sounds like the cerebellum has a pretty important role, right? But once scientists discovered this role, they thought, this brain region is probably good for motor skills only, and moved on. However, scientists are getting excited about this brain region once again, because new research finds interesting links between the cerebellum and autism, which is pivotal for our understanding of autism. Cerebellum functions differently in autism, and more than 90% of autistic children experience sensory signals differently, which may be related to a difference in sensory predictions of their cerebellum. Moreover, to many neuroscientists' surprise, this brain region is recently found to play a role in language and social behavior as well. For example, your cerebellum is actively working when someone forms unexpected sentences, such as 2 + 2 is apple. Your cerebellum is also helping you with mentalizing. That's your ability to imagine what someone else is thinking about. Given that sensory, language, and social challenges are part of autism, this brings up the question, could differences in the cerebellum explain autism? This is the big picture question of my research, and I'm focusing on the link between the cerebellum and sensory challenges as a first step. Using the MRI machine, I'm looking at cerebellar activity of autistic children while they experience certain lights and sounds, and I'm investigating how this activity relates to severity of their sensory challenges. By researching the once unfashionable cerebellum, I'm hoping to uncover how this brain region contributes to autism and come up with more targeted treatments for challenges in autism. And while fashion will always exist in science, it is important to recognize what drives these trends and turn our attention from what's fashionable to what's pivotal. Thank you.
Thank you so much, Melis. That was a phenomenal presentation. I'm honored to present the Certificate of Achievement for competing in the UC Grad Slam.
Thank you.
Now, let's just spend a couple minutes getting to know you. So what first sparked this interest between the cerebellum and autism?
I was actually at SFN, Society for Neuroscience meeting in my first year of PhD, and I went to a cerebellum talk and I learned about all these new things that we found out about that the cerebellum has been doing for us. And I didn't know. I thought cerebellum, motor control, that's all. And I was really excited about it. I remember talking to my friend from college, T1, and I was telling her, this is so interesting. And that interest stayed with me. And then I found a lab that was interested in looking at the cerebellum in the context of autism. And I was like, amazing. I want to understand this. And that's how my research started.
And so you must be working also across disciplines at UCLA within psychology and neuroscience. And
we're under the Department of Psychiatry, but yes, it's, my one of my mentors is a clinical psychologist and the other is a neuroscientist. So there's, a lot of interdisciplinary collaboration.
Ah, fantastic. What do you like to do when you're not doing research at UCLA?
Oh, I love singing. I also really like illustrating. I do some science illustration, so it's one of my interests within science communication as well. So
Does your cerebellum help with the singing?
Probably when I get the note wrong.
Okay, so this was President Drake's question. If you were to be UC president, what would be the first problem you would tackle with? But I could say you could be UC CFO. No, or do it about your campus. What would you like to see changed at the UCLA campus?
That is a big question for me to tackle today.
No worries, no worries. Yeah.
Maybe I will skip that question.
Yeah. Well, thank you so much. That was really wonderful.
Thank you. Thank you.
My pleasure to welcome our second contestant to the stage, Sasha Neil Pimento from UC Davis.
Have you ever walked to a particular room in your house and then wondered, wait a minute, why did I come in here? Then you struggle to remember what your intention was, laying out several possibilities in your head before you finally give in and conclude, I just came in here for nothing. A few minutes later, your phone dies while you're on an important call. And that's when you realize all you needed was your phone charger. Interestingly, this common occurrence is called the doorway effect. Now, while this happens to people who do not have memory problems once or twice a week, individuals with memory loss, such as those with Alzheimer's disease, experience the doorway effect several times in a day. This is alarming because frequent memory loss leads to increased reliance on family and frustration due to loss of independence. If we look at people aged 65 years and older, 1 in 9 have Alzheimer's disease, and this number only grows with increasing age. Because this is a significant healthcare challenge that impacts a large segment of the population, I have applied my domain knowledge in computer science to develop a technical solution that will help you remember why you walked through that door. To combat the doorway effect, I, along with my team at the UC Davis Alzheimer's Research Institute, have developed interactivewear, an intelligent smartwatch application that can be used to record audio reminders with the simple touch of a button. For example, using this application, you can record a speech note the moment you think of your phone charger. This speech is then transcribed to text and displayed as a reminder on your phone or computer in the form of a sticky note, which can always be referred to later on in case you forget your intention. Now, while the eyewear app can be used to reduce the occurrence of the doorway effect, its true power lies in the data it collects. I am interested in analyzing this data, like changes in speech patterns and mood detection. I can then leverage this data with other biomarkers, such as computer use and data collected from smart sensors installed throughout the home to detect cognitive and health changes, which can lead to early intervention and thus prevent crisis-driven care. Ultimately, my vision for this research is an integrated system that will improve dementia care while also giving Alzheimer's patients what they truly desire, an open door to a joyful and independent life for many years to come. Thank you.
Thank you so much, Sasha. That was an
Thank you.
excellent presentation.
Thank you so much.
There was a certificate of achievement for competing in the UC Grand Slam. Oh. Thank you.
Thank you.
I'm having a doorway moment. What am I doing here? No.
We'll bring in the watch
Yeah.
for you.
Well, if you don't have Alzheimer's, it's once or twice a day, right? Yeah. That really is phenomenal. Where are you in terms of, I mean, is this something you're developing to patent and license to work with iPhones and
Well, as of
Android?
now, as of now, our primary focus is to actually carry out the research. And for that, we are actually currently in the process. We have the app ready. We're currently in the process of pilot testing our app with patients. And based upon the data, we are like making changes because sometimes the UI may not be it may confuse the patients, or so we are working on that, as well as like we're collecting the data right now so that we can do the analysis, as I mentioned earlier.
wow. And you use it for very practical applications like, turning off the stove and things like that. But what about, could it also dovetail with medical advice and, make sure you're taking pills 3 times a day and things
Yes.
like that?
Yes, exactly. Actually, one of our main things is that even the vibration sensors, which I mentioned, we actually have that installed throughout the home, and it's actually used to detect whether a patient is taking their prescription pills, and that's based on the vibration patterns. So we know through that, we're able to monitor what exactly the patient is doing. And it's not actually camera sensors, but it's these vibration sensors that detect it. And through this, we can actually inform the caregiver who may be located remotely that, okay, this patient may not have had their pills today. So the caregiver can come in and be like, you have to take your pills. And then, this is what we are trying to do right now.
That's phenomenal. So have you always wanted to be in computer science, or what did you want as you were growing up? What did you envision yourself doing?
Well, as a kid, I wanted to be a Formula One race car driver.
It so happened.
But then in 4th grade, I crashed up at the go-karting course really bad. And I was like, I can't do this anymore. And it took me a while to learn driving. So I was like, this dream is out. And with regards to computer science, my mom was a software developer. And I think for me, my mom is like the biggest inspiration here. So I think that's when I kind of stepped into it, and I realized that, hey, I really like this as well. Mm-hmm. So that's what really made me step into computer science and go for it.
Terrific. And to Ms. Takuta's question, what do you plan to do with this after you graduate?
Well, after I graduate, I actually look to work in the industry, but at the same time, I'm looking to volunteer with Dr. Alyssa Weekly's research, who's seated here. And the reason is because I value her research so much, and I think it has taught me so much through my master's program. It has I think instilled in me a lot of skills and the ability to kind of think from the point of a patient perspective, where, you have to think about what will be easy for them. Because as a computer science student, we always try to make the platform fancy, we add icons, but these icons are not really intuitive to them. And I think that's what I want to do, is I want to be in the industry, but actually continue to volunteer so I can keep the door open to always going back into research.
Wonderful. Tell us a couple things that you like about UC Davis.
Well, there's a lot of questions today.
Yeah. Okay.
Well, I actually, I was kind of preparing my question for what you said. When you said,
you want to change? Okay. You can
No. I was preparing my answer to you when you were asking
Melis
about the what you would change about her UC. And I feel like I would not change anything about my UC, because I feel like my UC has been amazing for me. I think just in this journey to come here, They have all been so supportive of me, every single one of them. And just in general, like as a researcher, I just got my first research paper published, and then I came here, and I never expected to do this in my master's program. And I think the fact that I could do this in 2 years is because of the way the UC Davis system has been built and the research that they allow us to do. And I think I'm so grateful for that.
Congratulations.
Thank you so much.
Every fall, we always get emails from parents who complain that their child's dorm room smells of manure. They don't realize that UC Davis is a top ag school in the whole world. And so now I'd like to welcome our third contestant to the stage, Natalie Pino from UC Santa Cruz.
Everybody poops, and nobody likes to talk about it. But I do, because in my lab, the Cortez lab, we study diarrhea and the viruses that cause it. So why do we study diarrhea? Well, unfortunately, diarrhea is the second leading cause of death for children worldwide. And as you can see, it is widespread across the globe with particular concentration in areas with poor water access. One virus that causes diarrhea is called astrovirus. And while many of you may not have heard of astrovirus in name, over 90% of people are infected by it by the age of 5. However, because diarrhea is not as deadly in the US as it is in other countries, it often falls by the wayside. And in fact, only 8 labs worldwide study astrovirus, and we're lucky enough to have 2 of them at UC Santa Cruz. So what do we know about astrovirus? Well, scientists were able to figure out that because astrovirus causes diarrhea, it infects the gut, specifically the small intestine, which is the first region after your stomach. And if we zoom in real close to the small intestine, we'll find a 1-cell thick layer that's protecting you and your body from the onslaught of food, microbes, and pathogens that you put into it every single day. And that 1-cell thick layer regenerates itself every 3 to 5 days, which is mind-blowing. So what's protecting that? A thin film of mucus. That's what I study. That film of mucus is crucial because it's essentially your single line of defense. And somehow, astrovirus is able to get past that and infect the host. So what do we know about astrovirus and how do we stop it? Unlike other well-studied viruses like COVID, we don't know what astrovirus uses as its doorknob, per se, to get into the cell. We know it turns the cell into a little virus factory, and we don't know how it gets out of the cell without exploding it. That's where my work comes in. I have found that over evolutionary time, it seems like astrovirus is hijacking the specific type of cell that makes that critical mucus layer, and it's using it as a way to sneak out undetected by the host. So these cells are going about their day-to-day thing, making that perfect protection layer, and little do they know there's a virus in that mucus, and it's getting pooped out and passed on to the next human, making it a very effective and infectious virus. So ultimately, why should you care about this work? Well, by understanding a novel, widespread virus and how it works, we can better understand how to prevent it. And with this knowledge, we're actively working on a vaccine at UCSC that I'll be publishing on later this year. By unraveling the mystery of how this virus gets in and how it gets out, we can better understand how to stop it and save kids' lives. Thank you.
Thank you so much, Natalie. That was a wonderful, wonderful presentation.
Thank you. Photo op.
I personally know how important this is. I grew up in Ethiopia, and the infant mortality rate was really high there. And a lot of it was because of malnutrition and diarrhea. So having a vaccine, when I lived there, you know, there were things still like smallpox and other things, and vaccines have almost eliminated those. So congratulations on this,
Thank you.
on this important work.
Thank you.
Tell us about Santa Cruz. What have you liked about Santa Cruz, and what have you liked about your research lab there and the campus in general?
Yeah, Santa Cruz is beautiful. If you guys haven't been there, check it out. It's an incredible town. I love living there. We get to work literally amongst the redwoods. So out my lab window is beautiful redwoods. It's a wonderful place to work. And my lab, most of whom are here today, In person, I absolutely love my lab. My advisor is incredible. I love working for her, and doing this research is super empowering. So yeah.
And I read that when you're not doing research, that you like to climb and also teach children to climb. Tell us about that.
Yeah, so I climb at my local climbing gym. Shout out to Pack Edge. And I love climbing. When I'm not in the lab, I'm climbing, or I'm outside at the beach. And yeah, I teach kids how to climb. So yeah, I love teaching.
Yeah. Do you do any outdoor climbing? California has so many.
I'm starting to get a little more. It's expensive, so
Yeah.
gotta find friends that have climbing gear.
Yeah. And so is this an area that you always wanted to go into, or has this evolved over your academic career?
Yeah, good question. So my mom was diagnosed with ulcerative colitis, which is a inflammatory bowel disease, about 10 years ago. And so that, I think, got me interested in the gut and learning about the gut and her disease in particular. And then I had the opportunity to work in a colon cancer research lab in my undergrad. I did a summer at the University of Oregon, and I was super stoked on the gut and learning about how dynamic it is. And then now there's all these studies with the microbiome and the brain-gut axis. And so I think there's just so many cool questions to explore in the gut system. Yeah.
And then going on from here, academia or medicine, or what do you foresee?
Ideally, I'm hoping to teach at a community college. I love teaching, and yeah, I'd love to get to inspire other folks to pursue science and get excited about science. So yeah.
So tell us one thing that no one in your lab knows about you. This will be the first thing that they learn.
I don't know. I love my lab. Like, we are great friends, so they know everything.
Okay.
We're constantly talking. It's a fun space. So yeah.
Well, thanks again.
Thank you.
Congratulations. So now I'd like to welcome our fourth contestant to the stage, Onadi Oku from UC Irvine. Yeah.
I can remember when I got the call. It was just a few weeks after my sister and sister-in-law had gotten married. Hey sis, Britta's cancer. I said nothing at first. Cancer? You guys are supposed to be trying for a baby. She's too young for this to be happening to her. I finally found words. How can I help? Sadly, my story is not unique. I'd venture so far as to say more than a few of you in this audience have gotten that call as well. The field of oncology has made incredible strides in the past 50 years. We've gone from an overall 49% cancer survival rate to 68%. And that's fantastic. But unfortunately, up to 75% of cancer survivors will learn that their cancer battle doesn't end at remission. You see, in the last 30 years, clinicians and scientists have found that cancer treatments like chemotherapy, radiation, and the modern wave of immunotherapies cause long-term cognitive damage, with symptoms ranging from anxiety, depression, memory loss, and in children, as severe as a 3-point drop in IQ per year. Now, the cause isn't just the treatments themselves, but the immune response raised as a result. Think of cancer treatments as a potential instigator of an autoimmune reaction. No, I don't say this to deter anyone from seeking out treatment following their cancer diagnosis, because what if I told you that Hannah Montana was right, and we can have the best of both worlds? Kill the cancer while sparing the brain. Well, my lab does just that. If you can think of the autoimmune response acting much like a toddler, leaving nothing but destruction in its path, then our treatment works by preemptively putting that toddler on timeout. However, this is no easy feat. Since cancer treatments often involve unintended brain interactions, the first problem we faced was the site of injury, the brain. Unlike other organs, we can't just replace the damaged cells in the brain, making prevention key. Now, if prevention is key, that means we must intervene while cancer treatment is ongoing without jeopardizing the cancer-killing capabilities of these therapies. Now, my lab has successfully prevented chemotherapy and radiation from causing lifelong brain damage, all while allowing these cancer treatments to do what they do best, kill cancer. For my PhD project, I'm tackling this task with a modern wave of immunotherapies. And by the time I'm done with my degree at UCI, I hope to have wrangled that toddler and put him on timeout. My research offers cancer survivors the possibility of a return to normal life. It's too late for Britt. She lost her battle with stage 4 lymphoma in August 2021. But for the millions of Americans diagnosed with cancer each year, I still have the chance to help them. Thank you.
Thank you so much, Onadi. That was very, very inspiring.
Thank you.
And what important research you're doing. I've had many family members who've suffered and died of cancer. And I know my first wife was a UCLA professor or a UC Berkeley professor. And she said the thing that bothered her most about the treatment was what the impact it had on her brain and her research. So, so were you in this field before your sister-in-law was diagnosed?
No, I'm actually a neuroscientist by training, and for actually a couple years, I was a lab associate at UCI School of Medicine, primarily focused on traumatic brain injury research. But then after I lost my sister-in-law to cancer, I still wanted to remain in the neuroscience community, but then I shifted towards cancer research to honor her memory. And so while I did my rotations, my first year of my graduate program, I looked for a lab that would allow me to still do what I love, which is neuroscience, but also make a meaningful impact in a field that means a lot to me.
Yeah. And I imagine that the impact of chemotherapy on the brain is different from radiation on the brain, is different from immunotherapy on the brain. So you've done work in all three of those areas?
So for right now, I've done work mostly in immunotherapies, specifically immune checkpoint inhibitors. I'm also moving into radiation. So that's what I'll be working on for now. Other members of our lab are working on chemotherapy. So we're hitting every angle of cancer treatments.
Terrific. That's great. Yeah. So what do you like to do when you're not doing research on oncology and radiation?
Let's see. I do love to garden. And I also occasionally take like paint and sip classes. I am not very good at all, but I most recently painted a portrait of my dog, a little French bulldog. It came out pretty well, so I was pretty proud of myself.
Oh, that's great. Are you taking those on campus or?
No,
in
just
the
nearby in
the
the
community? Yeah. So, and what do you want to do with this? Or do you go on in medicine or research, academia?
Yeah, so I've primarily been in translational research, and after I graduate, I definitely want to pivot towards clinical research and possibly conducting clinical trials within the neuroimmuno-oncology field.
Wonderful, wonderful. And tell us a couple things that you like or dislike about UC Irvine, Zot, other than their.
One thing I love is it's literally 10 minutes from the beach, and Newport is probably one of the most beautiful beaches in California. Things that I dislike. This city, it's definitely a family-oriented city, which means everything shuts down by 10 p.m. But yeah,
that is true.
Other than that, I love the city. It's so clean. It's so beautiful. There's so much greenery. Yeah. Definitely recommend everybody to take a trip out there.
Well, thank you so much, Onadi.
Thank you.
And now, please join me in welcoming our fifth contestant to the stage, Victoria Chouvet from UC Berkeley.
Bacteria share the world with us, and their presence benefits our environment and our health. However, some of them can make us sick. When pathogens get into our body, they make more copies of themselves and they disseminate to various organs in the body. For example, the bacterium Listeria can travel to the brain, which causes a lot of damage. My main question here is, how do bacteria actually make it to their final destination? In my studies, I use Listeria as a model system, but the overarching goal of this work is to lay the foundation for future studies to understand how other lethal pathogens may get into the brain and how to prevent the spread. When I gave Listeria orally to a susceptible host, I found that bacteria made it into the liver and the spleen. Very few bacteria were ever found in the blood, but after about 2 weeks, millions of bacteria showed up in the brain. to get a better understanding of how Listeria actually makes its way from the gut into the brain, I use a mixture of bacteria that were labeled with a genetic barcode. Every bacterium is generally the same, but they have a small DNA tag that allows us to identify them. Using a sequencer, we can read the barcode on each bacterium and assess the diversity of bacteria in a sample. When I gave this labeled library to a susceptible host, I found that the liver and the spleen had a few barcodes within them. So there was a limitation for bacteria to actually cross the intestinal barrier. But when I looked into the brain, there was only one barcode. This means it's really rare for Listeria to make it into the brain. But once they are there, they can grow really efficiently. Previous work also suggested that the blood can work as a conduit to get bacteria into the brain. And in our work, the blood was poorly infected. So I tried injecting Listeria directly into the blood. When lots of diverse bacteria were in the blood, many barcodes showed up in the brain. This confirms that the blood can indeed transport Listeria directly into the brain. Overall, this analysis gave us insights into the critical points in Listeria's journey that can be targeted to prevent disease progression. My hope is that this work will serve as a blueprint for future studies to understand how other lethal pathogens may get into the brain, such as Salmonella and tuberculosis. Thank you.
Wow. Thank you so much, Victoria. That was fabulous.
Thank you.
Yeah. So what spurred your interest in microbes and the bacteria?
Yeah, I've been interested for a very long time in health and disease and how the sort of balance between the two and microbes and how they affect this balance between health and disease. And so over my scientific career, I've been in a few labs that have tackled different pathogens. So I started in actually bugs that only infect fruit flies. And then I went to malaria, and now I'm working with Listeria. So it's been sort of a journey moving forward.
Oh, that's fantastic.
Yeah.
You can help solve a family dispute we always have is we always get organic berries, and my kids, we have 6 kids, they wash them, and I don't wash them. I say it's better to not wash them. And,
You should wash them. Definitely wash them. You should definitely wash them.
I'll wash out pesticides, but I don't mind a little dirt. You know, it's like
I mean,
a dirty biome is a good thing.
I'm all for a dirty biome. Dirty biomes, wonderful. They do great things for you. But the Listeria can be bad. Generally, actually, we all eat Listeria. We probably will have it later today, and we'll all be fine. But in the few cases where it does spread to the brain, it can be very severe. So it's sort of, you have to be careful.
Okay, good. Well, hope none of my kids are watching right now. But so, yeah. So tell us about your time at Berkeley. What have you liked about the lab? What have you liked about the campus and the community?
Yeah, it's been really great. I'm actually finishing up, so I've been at Berkeley for 6 years.
Congratulations.
Thank
Wow.
you. Submitted my thesis 2 days ago. Very excited. Yeah, and so Berkeley has been really, really a great environment. The community there, and MCV in particular, has been incredibly supportive. All the people that have helped me throughout Grad Slam to like get me ready for this and have supported me the whole way. It's like in my PhD has been great. My lab is here. I met my husband there, who's also here.
Hey.
So yeah, Berkeley has been just really phenomenal for me.
That's great. And then, and what are your thoughts going forward? Academia or more research? Or
So definitely more research. I got a postdoc position. I'm French originally, and so I'm moving back to a postdoc at the Pasteur Institute. So I'm very excited for this.
And what kinds of things do you like to do outside of research?
Yeah, so my partner and I are really big skiers, and Berkeley is a wonderful place because you can take day trips. So that has been really great. And then I did karate for well over a decade, and there's a great karate community here. So if anybody's interested, I highly recommend. It's a great sport, does great things for you. Yeah.
Are you going to get back to Paris in time for the Olympics?
Yes, I will. I wish I didn't because it's going to be really messy.
Yeah, it will be really messy.
But yes,
But
no, I'm
historic.
really
It's
excited.
historic,
It's historic, and I'm hoping to go to a couple of events, so it will actually be wonderful.
Yeah.
I'm really excited.
Well, congratulations, Victoria.
Thank you very
so
much.
much. Yeah. Now, please join me in welcoming our sixth contestant to the stage, Iris Garcia from UC San Diego.
Imagine this, a weekend night out with some friends. It could be at your favorite restaurant, the bar, or even the club. The place is absolutely packed, so music and voices fill the room. You're trying to catch up and have a conversation with your friends, but you can't really make out what they're saying. The place is way too loud and way too crowded. Similarly, for our brain to work properly, it needs the right amount of volume and visitors. Luckily, it has the blood-brain barrier controlling what enters the brain from the blood. The blood-brain barrier, or BBB, is not just a physical barrier, but a set of properties special to the blood vessels of the brain. To help you better understand the blood-brain barrier, I want you to think about your brain as an exclusive VIP club, the brain. And the blood-brain barrier is like the bouncer to this brain, whom we'll call Bubba Bouncer. There are bouncers at other clubs, just as there are blood vessels all throughout your body, but none as exclusive as the brain. This bouncer takes on a much more active role. It controls the ins and outs of the brain, letting the necessary, like nutrients and oxygen, in, and keeping unwanted guests, like harm and bacteria, out. Holistically, the Bubba Bouncer makes the club thrive. Our lab studies everything about this Bubba Bouncer, in health and in disease. And interestingly, we found evidence that the blood-brain barrier may also be controlling glutamate levels in the brain. Glutamate is what brain cells or neurons use to talk to each other. A message gets passed from one cell to the next using glutamate. Thus, having just the right amount of glutamate is required for our brain to work properly. Having too little glutamate would be like having no message to send at all. And having too much glutamate would be like trying to talk in a club where the music is too loud. Uncontrolled levels of glutamate are associated with brain states like anxiety and other neuropsychiatric conditions. My data supports that the Bubba Bouncer escorts extra unwanted glutamate out of the brain. And this is super exciting because it suggests that the blood-brain barrier may play a role in preventing these conditions. Here's an example of applying this to medicine. In a person with epilepsy, there is often too much glutamate in the brain. What if we ask the Bubba Bouncer to work overtime and kick it all out? See the immense potential in targeting the blood-brain barrier for treating brain diseases? The Bubba Bouncer is, after all, whom you meet first to get to the brain. Thank you.
Okay.
Thank you so much, Iris. That was
Yeah.
really, I've never heard that metaphor before, but it's a great,
Good. Now you know. Now you know.
great analogy. How did you come up with that metaphor? Was it, did it come over time, or did it spring on you one in a big bang?
Well, I think it was a collaborative effort with my partner who's here. He is not in the science field, and he wanted to know what I'm doing and what I'm spending all day doing. So
we had to explain it.
We had to find a
Keynote.
way. Yeah.
Ah.
So I was like trying to say, oh, it's kind of like this. And he was like, oh, got like a bouncer? And I was like, yes. So props to him.
Okay, that's great. That's
Yeah.
great. And I understand the thing where the bouncer's not going to let too much in, but you're also saying that the bouncer can go in and weed out bad
kick 'em out.
actors in
Yeah,
the.
exactly.
Okay.
Mhm.
Okay. Good.
Yeah.
Tell us about your time at San Diego. It's
Yes.
one of our booming campuses right now.
Yeah, it's been wonderful. I'm in the MD PhD program there. So I've actually been in San Diego for like 6 years now, and I love it. It's getting better and better. We're getting a new train trolley
Non-stop
line.
trains.
Yeah. It's just improving. Great food, great weather, obviously. And I think that brings really great people to San Diego. So it's been great.
That's great.
Yeah.
That's great. So after this, you're doing MD, PhD, medicine, research, academia,
Yes.
Formula One driving.
Oh, that would be dang.
Ideally, so I'll finish med school.
Yeah.
And then I'll probably do some residency. I'm interested in maybe neurology or child neurology, something like that. And then hopefully after that, I could kind of like mostly have a lab and occasionally see patients as well.
Ah, wonderful, wonderful.
Yeah.
So tell us a couple things that if you were Chancellor Kosa for a day, what would you change about the campus?
About UCSD.
About UCSD, or is there anything? It's
well, the San Diego is getting more and more expensive to live in.
oh yeah, we know that. That's why we're building so much housing there.
Yeah. But I guess also like also related to the housing, there's no way to park on campus.
Yeah.
That's an issue.
And then what do you like to do outside of research and outside of your medical studies?
Yeah, I like to exercise. I've been getting into lifting. Just like, you know, something about like putting your effort into something and it like giving back to you, unlike research, like feels really good, you know,
Yeah.
like I put effort and I get stronger.
That's great. That's great. That's great. And had you always wanted to go into this area growing up, or had you started doing med school and then did the PhD, or?
I think I was really interested in science things in general, but actually neuro, I didn't do anything before coming to med school and realizing like the way that neurologists interact with their patients and like those types of diseases were super interesting to me. And then that's when I was like, oh, I totally want to study that in a basic level.
Terrific.
Yeah.
Well, congratulations. Iris.
Thank you so much.
Now, please join me in welcoming our 7th contestant to the stage, Ilana Bou Grossman from UCSF.
There are over 7,000 languages spoken in the world today. Think about a language that you don't know. Think about how it sounds. You might recognize some of those sounds from your own language, like the vowel sound ah or the consonant sound s. But what might be tricky is grouping these sounds together into the words they belong to. This is in part because, despite what you think, in natural speech, there are no clear pauses between words. In fact, there is no reliable sound-based cue that tells you how to group sounds into words at all. But when you're listening to a language that you can understand, this poses no problem for you. Those vowel and consonant sounds magically assemble, shoop, into words that now carry meaning. So how is it that we're able to so easily group sounds into words in languages we know? What changes when speech means something to us versus when it doesn't? I study these questions by turning to the human brain. Our lab has the unique opportunity to work with patients whose brains are being monitored by very small electrical sensors that a neurosurgeon places underneath the skull directly onto the brain surface. These patients, who speak many different languages, graciously volunteer to help out with my research by listening to speech in their native language and a language they don't know. While they do this, my lab mates and I record and observe the neural activity going on inside their brains. And excitingly, I found that you can tell, based on brain activity, when someone is listening to their native language. How? Well, by looking at those moments when one word ends and another begins, also known as a word boundary. When you're listening to a language that you can understand, word boundaries are effortlessly identified. You're doing it right now as you listen to me speak. And as it turns out, when I used machine learning algorithms to analyze the signals of the brain, I found that your brain is helping you to do this by tracking when word boundaries happen in speech, but only for languages that you know. So we concluded that an ingredient to spoken language understanding is our brains figuring out when words end and begin. This is one step towards demystifying the magic that gives rise to the uniquely human capacity for language understanding, something that we all possess. It's also an important reminder that when we encounter something that our brain does that feels magical, we can always pull back the curtain to reveal the complicated and fascinating work being done behind the scenes to make it all happen. Thank you.
That is so fascinating. Thank you so
you.
much.
Thanks.
So it's really interesting. It's not only the sounds, but it's also the word boundaries. So
Yeah.
if your language had a weird cadence or something like that, you could see the neural impact of that as well.
Yeah. And word boundaries look very different in different languages. So that's kind of a cue that you pick up on, which is fascinating.
Oh, that's great. How did you come to this interest in language and speech and?
Yeah, I've been thinking about that sitting over there. And I actually, I grew up going to a bilingual immersion school.
Uh-huh.
And it was a language that my parents didn't speak. It was Mandarin,
Ah.
Mandarin and English. And I think that general experience of learning a language from that young and kind of interacting with it through English too, brought forward this interest in language. And then when I got to college, I took linguistics classes and I absolutely loved them.
Yeah.
And so it was a great kind of obvious route.
Oh, that's fabulous. Do you find that people who speak multiple languages have more neural activity even in their native language, or is that not the case?
It's not totally clear yet. And actually it's something that I am working on now and hope to continue working on is multilingualism. So what happens when you have multiple languages that you're kind of dealing with? How does that change neural activity? It's something that's still ongoing.
Yeah. So if you're speaking Spanglish or something like that.
Right, you're
it's like
switching between languages, which is very common.
Exactly,
Yeah,
exactly.
exactly.
And going on from here, how would you see this research affecting, I mean, I'm sure with people with traumatic brain injury and recovery of language, this is probably very, very important research.
Yeah, I do. I mean, I think that this does have a translational impact at some point down the road, but I want to make a plug for basic science because I think that this isn't a really important thing that we get to explore without necessarily having a clinical application that we're focused on yet. But I think that it has it could have a lot of impact in the future. So, yeah.
And so do you want to go on in academia and research, or?
I'm not sure yet, honestly. The UCSF is an excellent institution and it has a great career and development office. And so I'm excited to work with them to kind of think through that process.
Oh, fantastic.
Yeah.
And what do you like to do outside of research here in beautiful San Francisco?
Yeah. I really like science illustration. So the slide was something that I illustrated, which was exciting and fun to do. And I also, as a San Francisco rite of passage, have started climbing. And I really enjoyed that so far.
That's great. That's great. One question I meant to ask everyone, do you guys pick your walk-up song or do they pick your walk-up song for you?
They pick it. They pick it. Although
okay. Next year, Pamela, we're going to change that. They get to pick their walk-up song when they come up here. So.
At UCSF, we pick our walk-up song, and it was pretty fun. So,
what was your walk-up song?
It was a song, I don't actually know, it's by Morphine. It's on language understanding. It was, very relevant.
Yeah. Anyway, congratulations.
Thank you so much.
Have a nice day.
Thanks.
And now I'd like to welcome our 8th contestant to the stage, Casey Ring from UC Santa Barbara.
I want to transport you all back to where my research journey began, reading Spillover. In its opening lines, it states, "The next big human pandemic, bigger than AIDS or the 1918 influenza, is likely to be caused by a zoonotic virus, one that jumps from a wild animal to infect a human host. Experts call such event a spillover and warn us to brace ourselves." Yikes. Published in 2012, Spillover is a chillingly accurate foreshadowing of the COVID-19 pandemic, leading me to the question that keeps me up at night. What causes spillover? And my initial thought process was simple. Zoonotic diseases require wild animal hosts, and wild animals need habitat to survive. So wouldn't some major habitat modification, like deforestation, initiate a spillover event? My PhD aims to test this hypothesis using Madagascar as a study system. And over the last 50 years, Madagascar has lost half of its natural forest cover to deforestation, resulting in the extinction of 9% of its endemic animals, which are species found nowhere else on Earth. So, with this seismic environmental change in mind, we went out and surveyed animals and their disease status across the deforestation gradient, beginning in the pristine forest, moving all the way to completely deforested agricultural fields. And these surveys focused on rodents, such as these endemic ones that look like little baby hedgehogs. And in my research, I found that as we lose our forests, we begin to lose endemic rodent species, which are instead being replaced by invasive rats. Ugh. And this is a problem because invasive rats are super mega bad guys known for hosting many diseases and often taking over disturbed habitats. So because of this, I found that invasive rats were the most significant predictor of disease in deforested plots. Additionally, invasive rats love humans, often living in close proximity, further increasing the risk of an accidental spillover event. And so we can see my research in Madagascar as yet another canary in the coal mine, warning us of the unintended consequences of deforestation, and further proving that our health as humans is deeply intertwined to the health of nature. Luckily, this knowledge informs prevention. To prevent the next big human pandemic, we must begin heavily investing in the conservation of our forests. It's been estimated that this would cost around $31 billion annually. And sure, that is a fat stack of cash. But it has also been estimated that we have already spent $15 trillion responding to the COVID-19 pandemic, making $31 billion look like a drop in the bucket. So instead of scrambling to clean up the mess, why don't we prevent the spill? Thank you. Thank you.
Thank you, Casey. That was a fantastic presentation.
Oh, thank you.
Yeah. I don't know if you've read any of the histories of the Obama administration. They were so
Talk about a canary in the coal mine,
they
am
were
I
so
right?
concerned about Ebola that
Yeah.
and just barely missing. That could have been, a huge pandemic.
Yeah, then the next president didn't do quite as good of a job. Yeah, exactly.
I'm also very interested in Madagascar. I'm on the board of the California Academy of Sciences, and we have a big project there, mostly focused on protein replacement because they, the lemurs and they're doing a lot with crickets and things there. So how have you found your time in Madagascar? It's supposed to be a beautiful, interesting country.
I mean, one of the most amazing, unique places I've ever been to in my entire life. Culture, community, animals, flora, fauna, all that stuff is amazing out there. But to talk about the protein thing is the thing that is driving the deforestation in Madagascar is severe food insecurity. Yeah. And so they're deforesting to provide room for agriculture. So they do slash and burn agriculture that creates room for rice cultivation. And so they need rice to survive. So the drivers of deforestation are really complex, and that's why it requires more. Kind of answering a new question I didn't even ask.
Yeah,
but
no, but
I
then
think
the
it's
protein,
important.
they go for wild game.
Yes, exactly. So that's, causes a problem there.
Yeah.
Yeah.
I saw on your the picture they shared that you were holding a snake on the department website. So
yeah.
What is a snake, and what's the story behind the snake?
Really, I call myself an amateur herpetologist because I love reptiles. Mostly, I like snakes because they're easy to grab and catch, but I am obsessed with lizards. And so that's what got me into science, just running around catching lizards, and then I ended up in disease ecology. So I just love them, looking at them, trying to run around and catch them.
Fantastic.
Yeah, I don't know.
Have you done any research on reptiles, or is that you said amateur? Okay.
Yeah, so I actually got my master's at San Francisco State, and my advisor's here, Andrea Sway. And the reason I got interested in disease is because the Western fence lizard is the primary host for ticks in California. And so I was looking at how their abundance, populations, distribution affect Lyme disease in California. And so that was my gateway drug into disease ecology was the Western fence lizard. Yeah.
Fantastic. Tell us about UCSB. What have you liked about it? What have you learned to surf yet? And,
oh yeah, I've learned to surf. I actually learned to surf when I was at my undergrad at UC Santa Cruz. So glow slugs as well. But yeah, so
take a wetsuit.
Oh yeah, for sure. Well, that's what I love about Santa Barbara. It's warmer. Our campus is like literally right on the edge of the coast. So I get to walk to the beach every day on my lunch break. My community there is really amazing. I love them. They're amazing people. Hang out, like all my best friends are in my ecology department with me. So I don't know if they're seeing me right now, but shouts out to you guys at home.
And a long history in that area. I mean, it's amazing.
Oh yeah, I love it there. Yeah.
Yeah. Well, congratulations.
Thank you so much.
Now, please join me in welcoming our 9th contestant to the stage, Shannon Brady of UC Riverside.
Okay, Shannon, you've got this. Just take a deep breath and turn around and give them a big smile. Hi. I was talking to myself. Do you all ever talk to yourselves? Yeah? Okay, I'm seeing some heads nod. Maybe it's when you're planning what to do later, trying to calm yourself down, or even letting loose a major expletive to blow off some steam. And despite what mainstream media might have us believe, this is actually extremely normal. In fact, children do it from a young age without anyone having to teach them how. And using self-talk is associated with better performance on things like puzzle solving, math problems, motor activity. Athletes are even coached to use it to improve their sports performance. Unfortunately, we see trends where self-talk use starts to go down around age 7, about the time kids enter school, most likely because others label it as weird or disruptive. In addition, there's almost no work investigating how self-talk might affect emotional experience. And this is important for child development because kids frequently have their goals obstructed through things like imposed bedtimes, prohibited items, having to wait patiently, and more. And all of these blocked goals can result in feelings of anger or frustration. Again, very common, very normal, but when they get too intense for too long, they can have lasting consequences, like worse academic success and poor peer relationships. So my dissertation aims to figure out whether self-talk might be beneficial for children's emotional management. How am I going to do that? First, I have to make some kids mad. I'm gonna invite children to the lab to participate in a few games, like this really great toy that's locked in a box. I'll give kids a set of keys and tell them that if they can unlock the box, they can play with that really great toy. Seems easy enough, but I'll let you all in on a little secret. The key to that lock is not on the keychain provided, so it's impossible to open. Children will be alone with this frustrating situation for 5 minutes, and during that time, I'll ask half of them to talk to themselves while the other half don't. Using video and heart rate data, I'll be able to see whether the children who talk to themselves more during report feeling better after, show fewer negative emotional expressions, and experience less physiological stress. If self-talk does prove to be beneficial for children's emotion regulation, this could be a game changer for child development. Instead of viewing it as weird, inappropriate, and something to be stifled, we can instead rebrand it as something helpful, natural, and something to be encouraged to help kids help themselves through big feelings. The best part is it's not just for kids, anyone can use it. So the next time you face a challenging situation, try talking yourself through it. You might just find the key to your own regulatory success. Thank you.
Thank you so much, Shannon. That was fabulous.
Good.
So we have 6 kids, and so I can tell you from both personal experience, I can affirm when self-talk peaked and the uses of it. I'm glad to hear that it's a positive thing because a few of our kids still continue.
I mean, don't we all?
I do it in sports a lot, but I tend to do it too negatively on myself instead of as affirmation.
I feel that as well.
Yeah. How did you get into this? It's so fascinating what you're doing.
Yeah, I mean, based on some of the talks I've heard today, I think it's true that there's a little bit of me search in everything that we do. So I noticed during the COVID-19 pandemic, when I was alone a lot, I was talking to myself and I was like, is that weird? Is that normal? Is that okay? So I did some research. I'm a child development psychologist by trade, and I found out it's very normal for kids to do it. It emerges spontaneously. It's very helpful. And so I started to go around to the people I know and I was like, do you ever talk to yourself? And everyone's like, no, of course not. And then a week later, they'd come back to me and be like, yeah, I do a lot.
That's amazing. Yeah. And how do you measure their emotional, well-being at the end? You're saying that the ones who talk to themselves actually feel better about themselves, even though they don't get the toy.
Yeah, that's what I'm hoping to find. So we do this in a couple of different ways. We take a multi-method approach. We ask kids to tell us how they're feeling. So in theory, the kids who do the self-talk might report less negative emotions. We also use the video recording to see what type of negative emotional expressions they might be displaying. And then my lab also looks at heart rate data, so cardiovascular information to tell us about indices of the parasympathetic and sympathetic nervous system. Ideally, we'll see the kids who talk to themselves more experiencing less distress and a little bit more physiological calming.
Ah, wonderful, wonderful.
Yeah.
I actually use it in traffic jams. Not talking to myself, but I sing little things and stuff, and it calms you down. So,
exactly.
yeah.
I mean, there's a lot more that you do than you might realize in your head, out loud. I'm more interested in the out loud self-talk because I feel like there's something special about hearing your words back to you. I heard a quote the other day that's like, how am I supposed to know what I'm thinking if I haven't said it out loud first? And I think it really goes hand in hand with what I'm researching.
Wonderful. Yeah. And you like Riverside?
Yeah, I really love Riverside. I'm a Bruin from my undergrad, go UCLA. But moving out to Riverside was really cool because you're actually prime location. You can go to the city if you want to. You can go up north to like the skiing areas if you want to. You can go to the beach. You can go to the mountains. So I really like having all the options.
Perfect. Well, congratulations.
Thank you
Shannon.
so much.
And finally, I'd like to welcome our 10th contestant to the stage, Amarish V from UC Merced.
Did you all know that whenever you eat a spoonful of yogurt, you're taking in millions of live bacteria that are beneficial to your health? Bacteria, along with viruses and fungi, make up the human microbiome, or what I like to think of as a community of organisms. And the largest reservoir of this community is our gut. And we know from previous research that this community is critical to our immunity, metabolism, and nutrition. I'm fascinated with a relatively unexplored component of this community, viruses, particularly bacteriophages, which are the most abundant viruses in our gut. Bacteriophages are viruses that invade and can destroy bacteria. They are the most abundant biological entities on Earth. And yet, most people have never heard of them. Some interesting facts about bacteriophages. Healthy people, shown here on the left, have a balanced bacteriophage population. But people with inflammatory bowel disease or IBD, shown here in the center, have an imbalanced bacteriophage population in their gut. Show of hands. I know it's already been mentioned earlier today, but how many of you are familiar with inflammatory bowel disease or IBD? Okay, a lot of us. IBD afflicts more than 11 million people worldwide and costs the American healthcare industry alone more than $30 billion annually. It is a lifelong disease noted for its chronic inflammation of the gastrointestinal tract, which is from your mouth to your rectum. It impacts every stratum of society to such an extent that many people have said, this disease runs my life. We currently manage IBD through a variety of medications, but there is no cure. What I want to know is, what do these bacteriophages do? How do they impact IBD? I'm answering these questions in Dr. Yuri Grass's lab at UC Merced, which examines how bacteriophages interact with our gut. So, am I doing this? I grow human gut cells prior to adding bacteriophages to them, as shown in the lower right-hand corner. I then infect the cells with bacteria and look for changes in cell behavior. And what I have found is that bacteriophages fight off the invading bacteria and thereby protecting the underlying gut cells from bacterial invasion and subsequent inflammation. This is really important because with my research, we can better understand how IBD is caused and also on how it might be treated. And this is so exciting because the impact of bacteriophages on human health has not been explored before. And this represents a truly novel area of research. My work will not only further some insights into an important component of this gut community, but will also help us understand whether bacteriophages are beneficial and help to alleviate disease or harmful and make it worse. In essence, are bacteriophages friends or foes? Thank you.
Fantastic. Thank you, Amarish. So that's really fascinating. So how do you differentiate between a positive bacteriophage and a negative one?
So through sequencing, they've identified patients with IBD have differential populations of bacteriophages, and some of them are even more highly abundantly present compared to healthy population.
So you look at the profile.
At the profile, and based on that, we identify what phages we can use in order to basically activate our gut cells.
And the graphic you have where it's fighting it off, can they differentiate in terms of which ones you're fighting and which ones?
Yes, absolutely. Okay. We can do that as well because they're specific to their bacterial hosts, and so we can differentiate which ones are actually causing an effect versus ones that don't.
Yeah, and what stage are you at in the research? I mean, is this going to be something that goes into clinical trials, or what's the?
So currently, my research is all in vitro, but there have been a couple of studies done in mouse work. So hopefully, we can extend this in mice, doing in vivo studies, looking at how bacteriophages directly impact gut health in that area. In terms of the field, there have been phage therapy studies where they've actually done this in patients. But some field is putting the cart before the horse. We don't even know what bacteriophages do to human cells. And so we're progressing before we even know the effects of it. And so this is where we get to explore what are the effects and are they harmful or not before we basically go into more in vivo work and then clinical trials.
Have you been able to use the new BSL-3 lab at Merced?
I wish.
That's a little plug for my own work.
No, not as yet. Not as yet. Well, I don't work with pathogens, so that's a differential.
Yeah. Tell us about UC Merced. What you've liked about it, and you're at the end of your studies, so you've been there quite a while.
Yes. Well, I'll be defending this summer. So yes, close to the finish line. Thank you. Close to the finish line. So I'm part of the QSB, or the Quantitative Systems Biology program, and it's an umbrella program for a lot of different areas of research. And so prior to joining my lab, I actually did rotations and got to know a lot of the faculty, a lot of the graduate students. And so it's a very, very integrative, tight-knit community. Yeah. And if I need, troubleshooting for experiments or just hanging out, there's a lot of company.
Yeah. Do you get up to Yosemite much?
I have. Yeah. I have. Not as much as I would like.
Yeah.
Sorry. But yes, but try to go as much as I can.
Well, congratulations. Thank you so much.
Thank you so much.
All right, what a incredible delight to hear from all of our competitors. These are really, really amazing presentations. So now I have the distinct pleasure of turning over the reins to my relatively new friend and colleague, Dr. David Rubin, who is the UC Executive Vice President for UC Health. Come up and join us, David.
Thank you.
Can I do your walk-up song?
So I just arrived like 6 months ago, but I get to say that I'm actually a graduate of the University of California, San Francisco, if that scores me extra points to start, right? Well, this was great. And I may just be back, but this event has been going on for about 9 years now, I believe, right? And it's really become this amazing showcase for the extraordinary research and communication skills of our graduate students across the University of California. And this year is no exception. There were definitely some themes here, though, right? We saw a lot of neuroscience. We saw some microbiome, some comparative medicine. I want to shout out to the veterinary school up at UC Davis, even though they weren't here today. Shows the value of that sort of collaboration across so many disciplines. I will say, though, I'm mindful as neuroscience seems to get a lot of the attention. It's like the popular kid in the class. For that graduate student out there who's studying the gallbladder, I see you. All right? And you could be up here next year, so don't give up, right? I will say that it also reminds me as I see sort of the crossover, computer science, basic science, psychology, development, all of the work here, just from my own career and just, we don't like to talk about the pandemic anymore, but it reminds me of just the amazing, when we break down silos and we get in a room with people from different disciplines, the power of that, and that represents the best of the University of California. And we saw that on the stage here today. I also am mindful of the power of communication. In my own careers, I've mentored and worked with others. I used to say to trainees and graduate students, think about what your mission statement is. We all like to think about research being a process by which you have a hypothesis and you test it, and then you get an answer, and then you figure out how to disseminate it. But no, each of you came here today with a mission statement that was based on your own lived experience. it's extremely powerful for telling that story. So even as you develop those research ideas, thinking about who your audience is going to be, who you're doing this on behalf of, and how you cultivate those stakeholders from the inception of your work, not only will help you when you deliver the actual science, but it will also engage your audience in a way that will ask tougher questions, for which you'll be able to test those in your labs. And finally, I'd say, there's a deeper truth that lies in the collaboration that we feel in this room today, that knowledge is not the sole possession of us in academia, but something that when we share it effectively can improve our human condition. These are touch points of light in a world that can feel, devoid of that at times. So let's celebrate our Grad Slam finalists, but also the spirit of our inquiry, our innovation, and inspiration, and all these relationships that make UC so great. All right, thank you. So I do have some assignments. We're going to go grab some lunch right now. And so I got assignment number 1, enjoy lunch. Enjoy it and meet each other and reach out to someone you don't know while you're out there. We're going to come back here at about 1 p.m. sharp, so don't leave. We're going to wait to see the votes. And then make sure you vote for that People's Choice Award, all right? You'll all have an opportunity to vote via text. Do that now or while you're waiting in the buffet line. The top voter is going to get awarded $1,000 for the People's Choice Award. Send a text to 1-800-400-2167. So 1-800-400-2167, and you just put the keyword in UC Grad Slam. And then note you can only vote once for those friends and family who think they're going to game it. You only vote once. Once you've submitted your vote, you can't vote again. Only cell phone numbers in North America can vote. That's interesting. I forgot the candidate, the one who's from Paris. Sorry, that's not going to help you, right? Unfortunately, this year, votes from international cell phone numbers outside of North America are not able to be accepted. Stay tuned to the event for the results. And so we've opened text voting. We'll keep it open for about 20 minutes. Enjoy the reception. Vice President Galat will be at the helm when we all return. And if you're in the building or watching online, please be back here at 1 p.m. as Vice President Galat announces the winners and Nathan presents the awards.
I want to say welcome back to our viewers here in the room, as well as those who are joining us online. My name is Yvette Galat. I serve as the Vice President and Vice Provost for Graduate, Undergraduate, and Equity Affairs for the University of California, as well as its System Chief Diversity Officer. And I want to add my appreciation and thanks to, and congratulations to all of our students for the impactful research they are doing and their outstanding presentations today. And I want to echo Dr. Rubin for appreciation for how much you are in conversation with each other through your research. It was just phenomenal to hear how much is in sync. And so I think the future of our brains, our bellies, and our biodome is in really, really good shape. So I hope that others see through you the possibilities in their own life for future research. It was just wonderful, wonderful. So now is the portion of the program where I am the most popular person in the room. And why is that? It's because I have the great honor of announcing the awardees of this year's UC Grad Slam competition. And that is exactly what I'm going to do. But first, before we do that, you're going to have to wait just a little bit longer. I want to recognize and thank the graduate deans from each of our campuses. They're responsible for leading graduate education, and they've made this day possible because their campus competitions have been coordinated in such a way to bring us the outstanding competitors that you met here today. They not only host their event, but they commit to doing this annual system-wide tournament as well. So for those who are present, what I'm going to ask you to do when I call your name is feel free to stand. And I would ask that our audience collectively acknowledge you after I've announced all of you. Lisa Garcia Bedoya from UC Berkeley. Jean-Pierre Delplanque, JP Delplanque from UC Davis. Jillian Hayes, our graduate dean from UC Irvine. Susan Etner from UCLA. Harant Karachian from UC Merced. Lydia Cos, UC Riverside. James Soto Anthony from UC San Diego. Nikkei Blake from right here in San Francisco, UCSF. Lila Root from UC Santa Barbara. And Peter Beal from UC Santa Cruz. So one last round. Stand, stand, stand. Thank you very much. And now it is time for the awards. We're going to start by announcing the People's Choice Award, the third place, then the second place, and then the grand prize winner. And the grand prize winner receives the Slammy. For those of you who are new, the Slammy is a distant cousin to the Grammy, so it is part of a very prestigious awards family. So you all have to stay tuned to find out who wins the coveted Slammy. So as you all know, these were incredibly impressive finalists. And I know our judges had some hard work to do. So let's hear from our audience judges first. Our thanks to everyone who took the time to text the 800 number and share with us who you thought did the best. And we're going to announce the system-wide People's Choice Award. And this individual will receive $1,000. All right, so Nathan, do you come on up? Perfect. Yeah, you got the bag man has to be on the stage. All right, here we go. All righty. The 2024 People's Choice Award goes to Casey Ring from UC Santa Barbara. And graduate dean Lila Root. Congratulations. Oh, okay. Congratulations. Thank you so much. All right. Now it's time for the third place winner. The 2024 third place winner is Shannon Brady from UC Riverside. Lydia Koss is our graduate dean. One turned around. Congratulations. I messed up. We're going to take a short pause. Everyone talk to yourselves. Talk to yourselves. No la la land. No la la land. Why are we going to say? This is unprecedented in Grad Slam history. The 2024 second place winner is Casey Ring from UC Santa Barbara.
now this is really exciting. I don't even know what I'm supposed to do now. No, I'm kidding. I'm going to resist my very strong temptation to artificially build drama because we just had it. There is only one thing left to do, and that is to name the UC system-wide Grad Slam winner. And the Slammy goes to Iris Garcia Pat from UC San Diego. Bouncer. Now we have another tradition in Grad Slam, which is what we call the perpetual plaque, and it moves from campus to campus with the graduate dean recognized for producing the winner of Grad Slam. And this year's perpetual plaque will go to Dean James Soto Anthony from UC San Diego. And if you're here, James, I don't think you are here, but we will add your name to this plaque and you will be able to display it in your office or wherever for the full year. So congratulations. And that brings us to the conclusion of our program. And so on behalf of President Drake, our intrepid CFO, Bagman, Executive Vice President, Nathan Brostrom, and all of us at the Office of the President, we want to thank our partner, LinkedIn. They have done a marvelous job again this year in supporting us. We appreciate your continued support and for working with us to present this event. It is so important to us. I want to give a very special thank you to LinkedIn Talent Solutions, which is the production team, the audiovisual team, and the event personnel. We appreciate you. Thank you so much. And of course, as you know, Grad Slam would not be possible without the help of so many people. There are far too many for me to mention by name, but I do want to give a shout out to our entire UCOP Grad Slam team. Stand up. And then a very, very special thank you goes to our graduate studies team at the Office of the President, headed by Pamela Jennings. And to our marketing and communications team, who are always our vital partners for bringing you the magic of UC Grad Slam. We appreciate all of the energy and all of the intellect that everyone brings to this event. And finally, I want to thank all of you for joining us for this very special event. We appreciate your wonderful support of our talented students and the great energy you have shown us on behalf of graduate education at the University of California. We've given you just a glimpse of what graduate education is like at the University of California. And I hope that you will take the inspiration and the insight from today and help find ways to actively support graduate student research at the University of California. Funding, advocacy, and engagement are so important to us for supporting graduate education, and graduate education would not be possible without that support. So it has been a pleasure today to share time with you. Thank you so much, and we will see you next year.